the two DNA pieces coming from two distinct genes are put head-to-head and are read by messenger RNA as one messenger RNA encoding a new fusion product called the PCR-Abelson protein, which has a very strong constitutive tyrosine kinase activity, which is really the oncogenic factor for chronic granulocytic leukemias. You know that for tyrosine kinase, which is sponsoring phosphor ions, but it's important to have a phosphor donor, which is ATP. And ATP is the binding site of this molecule, and this is important to release one phosphor on a tyrosine substrate on another molecule here to achieve this tyrosine kinase function. But by knowing the structure of the gene, you can identify the ATP binding site. By knowing the structure of the gene and the structure of the protein, you can have a 3D crystallography of the protein and identify the 3D form, the 3D shape of this ATP binding site. And then the cancer biologists can ask the chemist, please, would you design for me a new drug which would be able to bind exactly the shape of this ATP binding site and which would be a competitor for ATP towards its potential binding site on the molecule. And it's exactly the history of Gleevec. Gleevec has been discovered by chemists, but under a design which has been proposed and suggested by cancer cell biologists. And Gleevec is acting as a competitive inhibitor of the binding of ATP on the PCR-Abelson molecule. So you see that the process is completely reversed by the fact the knowledge of cancer cell biology is allowing the discovery of new important targets. You know that Gleevec was also shown to be effective in this strange gastrointestinal sarcoma tumor called GIST, not by acting on Abelson, another activated oncogene, which is c-KIT, which is expressed on this tumor. And those tumors, which was extremely resistant to any kind of chemotherapy or any kind of radiotherapy, only treated by limited surgery, but with very short, very bad results and with relapse after surgery, with a very low life expectancy, are extraordinarily treated by Gleevec, and the life expectancy is much longer with Gleevec treatment. So these are very extraordinary stories, but it opens the way to identify new targets. But this is a huge task because the progress in cell biology has really helped us to identify multiple targets, either in the form of binding cells of this receptor, or secondary signals in the cytoplasm, or DNA binding elements which regulate expression of genes. And all of these new targets can be potential targets for anti-cancer drugs. So many laboratories and maybe a number of natural products which could not be identified as real cancer drugs by conventional means, and maybe a number of products of traditional Chinese and Oriental medicine could be actually good targets for one of this pathway and could be tested of these different targets. But now the problem is really complicated by the number of new targets, the number of new compounds, and the number of new trials we should do. And you have to remind that if you have 100 new targets and 50 potential inhibitors of each target, you can predict, but only by screening them one by one, it will be very long to treat and cure cancer, because less than 5% of European patients are entering clinical trials after cancer. They are treated by conventional therapy and not in clinical trials. The duration between the entry of the drug in clinical research and entry of the market is, in the good terms, 5 years, but in average 8 years. And tumor response are assessed by very rough means, which are mainly clinical radiology, tumor measurements, which are actually poor criteria, and we need new criteria to assess response to treatment. And then you have to understand that all this, the clinical story could be unique, because probably all these drugs will not be sufficient to treat cancer, which accumulates genetic events and will accumulate targets for new drugs. So targeted drugs will be more efficient in combination than even one by one. So if we mix all these parameters, we see the importance of cooperation between institutions and between countries, because testing every potential target with every compound in preclinical and in clinical practice and combination of potential drugs will be extremely long. And I think it's really the beginning of a new era and for making so important international cooperation. I will discuss later this afternoon of other aspects on which, because of translational research, we could facilitate and strengthen